Hey biology students! In this biotechnology video, we're going to describe the process of DNA profiling. So I'm going to start by having you watch a short video clip that walks us through the steps involved in DNA profiling, and then we will talk about what you viewed in the video.
These 5 men were the prime suspects in a murder case. Not long after this picture was taken, the investigators found that numbers 4 and 5 in the lineup had legitimate alibis. The forensics team scoured the crime scene for evidence to use in DNA profiling, which could help determine if one of the remaining suspects is guilty. But what is DNA profiling and how does it work? DNA profiling is the use of variable parts of the genome to detect genetic differences among individuals. The technique is especially valuable in criminology because it allows investigators to match DNA found at a crime scene with DNA from potential suspects. DNA may occur in blood, saliva, hair roots, perspiration, semen, and other substances left at a crime scene. In this case, the forensics team found a small blood stain on the victim's jacket. They want to test the possibility that the victim drew blood from the killer, perhaps during a struggle. The investigators begin by extracting DNA from the blood stain. Every white blood cell in the sample contains 1 copy of each of the 46 human chromosomes inherited by the blood's owner. Sequencing the entire genome would be wasteful, because everyone's DNA is about 99.9% identical. Instead, the scientists analyse only selected regions that are known to be extremely variable among individuals. The most common approach to DNA profiling is to examine short tandem repeats, also called STRs, which are sequences of a few repeated nucleotides. The STR example shown here has repeats of the nucleotide sequence AGAT, but the exact nucleotides depend on the STR site. Each STR locus has many possible alleles, which vary in the number of repeats. For example, the STR region on chromosome 21 has anywhere between 12 and 41 repeats. DNA profiling counts the repeats at the 13 STR loci in a person's genome. Unfortunately, the amount of DNA found at a crime scene is typically too small to analyse directly. Instead, the DNA at each of the 13 STR loci is amplified using a technique called the polymerase chain reaction, abbreviated to PCR. PCR makes many copies of a DNA sequence. Here, PCR is making many copies of the DNA at an STR locus from one version of chromosome 5. PCR also amplifies the same STR region on the homologous chromosome. After PCR, the scientists have enough DNA to begin generating the DNA profile. We'll focus now on the amplified STR region of chromosome 5. A technician determines the number of repeats at the STR site on both versions of the chromosome. In this case, the STR region on chromosome 5 has 7 repeats on one chromosome and 9 repeats on the other. The blood sample's genotype for this STR region is therefore 7-9. The scientists use the same technique to count the number of repeats at all of the STR sites. The result is a DNA profile that lists the genotypes of all 13 STR loci in the blood sample. Investigators also determine the DNA profile of each of their suspects. The probability that any 2 unrelated individuals have the same pattern at all 13 STR loci is 1 in 250 trillion. So a perfect match is convincing evidence that the DNA in the blood sample actually came from a suspect and not another person. This time, DNA profiling solves the case. The DNA profile of the blood sample found on the victim's jacket perfectly matched the DNA profile of suspect number 2. Case closed. DNA profiling has also been applied to biological evidence collected during past criminal investigations. Since 1989, DNA profiling has led to the release of hundreds of wrongfully convicted people, many of whom were serving time on death row. Beyond solving crimes, DNA profiling also has many other practical applications. For example, a court may order a DNA test to determine whether a man is the father of a particular child. DNA profiling is also used to determine if twins are identical or fraternal. Overall, DNA profiling is a fast and inexpensive application of biotechnology that takes the guesswork out of criminal cases and family disputes.
Okay, so now let's talk about what you viewed in the video. So here we're looking at the STR loci for these three individuals, and we'll go ahead and call them suspects because one of the applications, as you learned, of DNA profiling is to identify the perpetrator of a crime if DNA evidence was left at the crime scene. So here we have these three individuals. So we'll just call this guy with the glasses, he's going to be suspect number 1. This blonde woman is going to be suspect number 2. And the red-haired gentleman is going to be suspect number 3. Decide which of these three you think look the most guilty. Okay, so then we're looking at the STR loci on 2 chromosomes. They're homologous chromosomes because, of course, we have 2 copies of each chromosome. We are diploid. So recall the STR refers to short tandem repeats. So on particular regions of our chromosomes, certain chromosomes, we have these STR repeat regions. Recall these are highly variable between individuals. And when we look at a collection of 13 of the STRs, which is what the FBI uses in their database called CODIS, then it creates this profile. So here we're looking at the repeats of A, G, A, T, and we see that these 3 suspects have different combinations of these repeats. So suspect 1, his profile would be 7 repeats on one chromosome and 6 on his other. Suspect 2 would have 4 repeats on one chromosome and 19 on the other. And suspect 3 would have 12 repeats on one chromosome and 14 on the other. So we can already tell they have a very different genetic profile for just this one particular STR loci. So in the video, we learned that the next thing you'd want to do would be to do PCR. So PCR is a technique that's very widely used in biotechnology. It stands for polymerase chain reaction. And it was discovered when a scientist realized that there is a heat-stable polymerase called Taq polymerase that can be used in this reaction. So it's a DNA polymerase that has been isolated from a microorganism that actually lives and thrives in hot springs, hot water. So it's heat stable, and that's what's used in this reaction because we actually heat the DNA and cool the DNA and repeat heat and repeat cool over and over and over through all of these cycles. So what happens is you have your target DNA sequence, and the heating purpose is to denature the DNA or unzip it. So we would unzip the DNA, and then if you have your particular target sequence that you want to amplify, you have these little short segments we call primers. So we would have a primer that would anneal here, And we would have a complementary primer that would anneal here. And this is achieved by cooling the mixture in the test tube. So you heat it. A PCR cycle involves what we call the denaturing step. And so you heat the mixture to 95 degrees, and the purpose is to unzip the DNA. That weakens the hydrogen bonds that holds the two strands of the DNA together, so that unzips them. And then we have a cooling step, we call it the annealing step. So we cool to usually around 65 degrees Celsius, and this allows for attachment of primers to the target sequence. So the primers are very specific to that particular sequence. Okay, so you have your target gene. So in this case, our target gene would be in this region. This would be our target gene that we're going to try to amplify through this process. So you cool it, the primers will attach to the target sequence, to the target, we'll say target gene. And then the cycle will begin to heat again. So we have what we call this the extension step. And so what happens here is you slowly heat to maybe 75 degrees or so. And what that's going to do is that heating is going to speed up the process of DNA replication. So you're going to get this DNA polymerase, this Taq polymerase. We call him Taq polymerase, but he's a DNA polymerase. We'll call him Taq polymerase there. And so he will extend the primers. So he'll start building complementary DNA. Let's do another color. So it comes along and starts building the complementary DNA sequence, extending the 3' end. Remember, DNA replication is always 5 to 3' direction. So we're moving in an arrow going this direction from 5 to 3'. And the polymerase enzyme would be adding the nucleotides as it moves along. We'd be going in this direction on this strand from 5 to 3'. So adding those complementary sequences. So at the end of one cycle, you end up with 2 DNA molecules when you started with 1. So now you have 2. But the beauty of the polymerase chain reaction is that you run this in a machine called a PCR machine, and it just repeats these cycles over and over. You can program the machine to run a certain desirable number of cycles. So 2 to the power of 35, let's say we want to do 35 cycles. So you just set this all up in little test tubes. You put this mixture that includes your DNA that you collected, say, from the crime scene, and you might just have a little bit of DNA at the crime scene. You might just have DNA that was left on somebody's clothing or chewing gum or a cigarette butt. You know, you might have something like that. Somebody just chewed on it and you have a little bit of their DNA. Well, in order to do an accurate DNA profile, this is why we're doing PCR. We're doing PCR so we can amplify the DNA. In other words, make millions of copies of the DNA molecule. So the purpose of PCR we say is to amplify the amount of DNA. And usually that's also, we're looking at a specific gene. So we might be looking at an STR, we might be looking at a gene involved in a mutation, we might be looking at a particular gene that's an identifying sequence for a particular strain of microbe that we want to identify. So you run this through the cycle, it goes 35 cycles. So that's 2 to the power of 35. How many, what is that? Well, let's plug it into a calculator. 2 to the power of 35. 34 billion. So now we would have 34 billion copies of our gene that we amplified through this process. Approximately 34 billion copies. Now we have a lot of copies of our gene of interest, of maybe that STR loci. So in the video, she shows you one way to look and see how many repeats somebody has is to have a computer readout of that. There is an alternative though. It's called gel electrophoresis. And if you take the lab portion of the course, if you're in my Bio 100 instead of my Bio 101, we do this lab. Well, we're not doing it this semester, are we? But guess who did? I did. And I posted a demo of it. And I'm actually going to show you guys part of that demo because everybody needs to understand this technique. So gel electrophoresis, the purpose of this technique used in a biotechnology lab would be to separate specific lengths of DNA by size. And so we do this by using something called a gel. And you pour it, just, it looks a lot like Jello actually. It's like making Jello, you pour it into a mold, and it's this square or rectangular gel, okay, and it actually is called agarose. So it's made out of something similar to agar, which is a gelatin material derived from seaweed. Anyway, so when you pour it into a casting tray, you actually end up with these little wells at the top of the gel, and this is the loading zone for where you're going to put your DNA samples. And so we're going to load the DNA in each well. So for each sample that you're running, you load the DNA into the well. So we'll put our DNA in there. And then notice in this diagram, they're showing us that we connect this box, this gel box, to a power supply. And you do, and so you connect it to a power source. So what happens is there's a negative charge at the top of the gel and a positive charge at the bottom of the gel. We call this side the anode. The negative charge is the anode, and it has a black lead. It looks a lot like your car battery. If you've ever taken a look at your car battery. So you have a black lead leading to the negative anode and a red lead that leads to the positive cathode. And so we hook it up to this power supply, and it basically sets up a battery. So it's going to cause an electric current to flow through the gel. And it's usually in a box where there's some water in it. It's like an electrolyte bath that you add to the gel box. And that allows for the electric current to flow. And you see bubbling when you see this current flowing through the gel. And what happens is, because DNA is negatively charged, and that's because, if you recall, DNA is composed of this sugar phosphate backbone, and then of course it it repeats on the other side. So what happens is, and then of course we get this connection happening. This continues between the base pairs. We get the connection. So recall the phosphates. So the phosphates are negatively charged. So that actually is important to remember because that's going to explain why the DNA actually gets pulled towards the positive or the cathode end when this current starts flowing through the gel. So what happens is the DNA starts to migrate through, and if you have DNA of different lengths, then they're going to show up as these bands that start migrating. Let me see. Migrating through the gel, like so. Okay, and so we call these bands. And what happens is the bands, they represent segments of DNA, like for example, maybe segments from the STR loci that you just amplified by PCR. And it makes it so that we can actually see the fragments show up, okay? But the way this works is that the shorter the length of the DNA, the faster it moves through the gel. So that's a principle of diffusion, actually. The smaller the molecule, the faster it moves. So that's actually how they separate. Let me erase all that. So the principle is then at the top of the gel, we see the longest DNA fragments. They're longer, so they don't move as fast, so the longer fragments would be at the top, and then sort of medium size in the middle, and then the really short DNA fragments would be at the bottom. Okay, so top of the gel we get longest, bottom of the gel we end up with our shorter fragments. Now I'm going to let you watch a sped up version of that lab that I do for Bio 100 of gel electrophoresis, and you'll watch me load the gel. So here you're going to watch me load the agarose gel with the samples from this experiment, and you have to carefully add the samples using the micropipette transfer tool into these wells. Once I've added the samples, I'm showing you the samples are there, then I put the cap on it, and it has these electrodes. So the black electrode is the negative electrode or the anode, and the red electrode is the positive cathode. So I connect it to the power source, and then the bubbles start to flow. So this has been time-lapsed over about 15 minutes. We're seeing migration of these DNA bands through the gel matrix. And the DNA is attracted to the positive end because it has a negatively charged sugar phosphate backbone due to those phosphates, if you recall. Okay, so now let's practice gel electrophoresis with our 3 suspects from earlier. where we know what their repeat numbers would be. So imagine we were going to run these onto a gel, and we have 3 samples, so we'll say we have 3 wells. And remember, this was suspect 1, suspect 2, and suspect 3. So usually what you do is you usually have some sort of reference control, so you know how big certain, where the bands would actually land. Remember, we always say we move from negative to positive end of the gel, and let's say our reference is going to be, we'll say 30, 25, 15, 10, and 5. This is going to be for number of repeats. Reference spot. Okay, so we have suspect 1, suspect 2, suspect 3. So let's see here. Suspect 1, you have a 7 and a 6 as those bands that would show up. So we might have a 7 here and a 6 here. Suspect number 2 has a 4 and a 19. So the 19, here's the 25, so the 19 would probably migrate somewhere around here, and then the 4 would be a little bit smaller than the 5, so probably down in here. Now what we're really looking at is the placement of the bands. Where are they placed in the gel? So I made these a little too short, so I might lengthen a little bit, but actually the length here doesn't really matter. Neither does the thickness of the band. Isn't really what you're looking for. What you're looking for is the placement. Where is it placing? Okay, suspect 3, you are at 12 and 14. So that would probably be the 14, and then that would probably be the 12. Okay, so that would give us a reference sequence to compare to the crime scene. So if we had another lane where we ran the crime scene, DNA from the crime scene, so let's add another lane. We'll call this crime scene. So the DNA from the crime scene, let's say that ended up looking like this. What we would do is we would use that pattern to look to see who of our 3 suspects also has that pattern. So we're looking for this band here. Well, suspect 2 has that band. Who has this band? I didn't do very good in my lineup, but we're going to say it's that band there that matches. So we would say suspect 2, she looks the most friendly, but she would actually be as the DNA that matches the crime scene. Usually we look at multiple SDR loci, not just the one, because the more loci you look at, the more statistically reliable it is. How about this one? So here we've got the crime scene, we have the victim's DNA profile. Sometimes that's also included as well, just in case the victim's DNA contaminated the crime scene DNA. But same sort of thing, we look band to band, where do we see the match? So it looks like we're seeing band for band a match through suspect 2's lanes here. Different crime, but there we go. So we would say we have a match here as well. I thought I'd give you one application to current situation in our world right now, an application in biotechnology, and it's called RTPCR. So pictured here, this is the infamous SARS-CoV-2, which is the strain of coronavirus that's responsible for COVID-19. So COVID-19 is the disease, and this is the strain, SARS-CoV-2. So this is what the virus looks like. This is a schematic, a cartoon image, but showing us what this virus looks like. And you've been hearing in the news about testing for COVID-19. How are they doing this testing? And the testing has been taking a couple of days, although they just released a rapid test. But I want to show you this one. This one is actually fairly accurate, maybe the most accurate test for determining if somebody is infected with SARS-CoV-2, and it's called RT-PCR. So we already learned about PCR, so what is this RT-PCR? Well, this is used when the sample is RNA, not DNA. So the thing about coronavirus is that its genome is RNA. Now, viruses are weird because they're not cells. They're technically not even alive because they fail the definition of life in biology because they're not cellular. And it doesn't have DNA in its core. Instead, it has an RNA core. So it has an RNA, what we call RNA genome or RNA core. So it's referred to as an RNA virus. And so every viral family has its variation on this theme. Some of them have DNA, others have RNA, but they never have both. They have one or the other. So coronavirus family, they have an RNA genome. So if you want to detect if somebody has this virus in their respiratory system, well, the first thing they're going to do is they're going to do a big swab. So they're doing a nose or a throat swab to collect some viral cells that should be in that area because this is a virus that invades those cells in the respiratory tract. So you would start with a nose throat swab to collect. So we would be collecting viral particles. So you would pick up some of the patient's cells in the process, but we would want the viral particles. And then we would do RT-PCR, and this would be to detect specific genes, specific SARS-CoV-2 genes, but because it has an RNA genome, so it has an RNA, that PCR only works on DNA. So that means that there's an extra step before PCR can be performed. And what happens is they take that RNA and they use an enzyme called reverse transcriptase, which has also been isolated by a virus and is used in biotechnology. That's a cool enzyme name, I think, because it does the reverse of transcription. So we've learned about transcription in this unit. Well, it does the reverse of that. So the reverse of that would be to go from RNA to DNA. Remember, transcription, you move DNA to RNA, so reverse transcription is the backwards version. So the viral RNA is is what you use, but then you mix it with the reverse transcriptase, and then that reverse transcriptase will actually make a DNA copy. So it'll copy it into DNA. Okay, and so that way then you can use the viral DNA to do PCR. And then you can use the PCR and then the gel electrophoresis to detect the specific genes involved. And so the specific genes that they're detecting several genes involved in the nucleocapsid. So they call them the N genes. So there's the N1, N2, and N3 genes. So these are part of the test. And then they also use it to detect a specific gene involved in RNA copying. It's called an RNase. It's called RNase P. So they are detecting these 3 different genes by PCR and then by gel electrophoresis. But this is something that takes a couple days, minimum of like 2 days really, to do it in a laboratory to get that information. I wanted to show you actually what that looks like, what they're detecting. I found this on the FDA website. So here, this is from the FDA website talking about the COVID-19 RT-PCR test. So it tells us we're going to detect the nucleic acid from SARS-CoV-2 in upper and lower respiratory specimens, such as a nasopharyngeal or oropharyngeal swab, sputum. They can take lower respiratory tract aspirates, and other stuff that people might be coughing up, basically. They can detect the virus in there. Okay, so I thought this chart was probably the most interesting. Here they're telling us about the RT-PCR results and how they can read those results. So, remember I said it was the N1, N2, N3 plus the RNase P gene they're detecting. So if somebody tests positive for all 3 of those genes, plus or minus the RNase P, then that is a positive result for detecting SARS-CoV-2. Report that to public health, it says. And then if only one or both of these targets are positive, but you get plus or minus on these, then that's also a report to public health. Then we have presumptive positive, which means they need to do an additional test because there might be some cross-reaction with another type of virus that could cause the same type of symptoms. And then they show us what a negative result looks like, where if you just test positive for the RNase P, that could be found in other viruses, so that's not specific to this particular virus. And then if you get no reactions, then it's invalid. So here's an application using biotechnology, and biotechnology is in need. It is in demand right now with our situation. Finding tests that are faster and more reliable to detect COVID-19 in individuals who are showing symptoms, and then also to develop medications to treat this infection, understanding the viral life cycle in order to design pharmaceutical medications that interrupt stages of the replication process of the virus, / now we have the vaccine race. So many different groups of scientists are racing to get a vaccine developed for this virus, and that is going to be a race. They're going to try to do it within a year, and that is really pushing the timeline to get that done. Anyways, I hope that was interesting, and we'll talk to you later.